Sharks. We know them for their fantastical and fearsome reputations. If a shark stops moving, it will die. Sharks are the apex predator in the ocean, top of the food chain in fact, capable of smelling blood from a mile away. Or even that sharks don't get cancer. You've probably heard some variation of these. But you'll be disappointed to hear just how much of their reputation is mostly tall tales. Not only are most of these tall tales over-exaggerated, but some of them simply aren't true. For example, while cancer is certainly less rare in sharks, it is not unheard of. But even if sharks aren't exactly how they are portrayed in the movies, they are still a medical marvel. While sharks are known to get cancer, they are also known to be resistant to certain kinds of cancer and even diseases we struggle with. Scientists have found that sharks are even more naturally resistant to COVID-19 than we humans are. But why are sharks so much more resistant to disease? Well, before I answer that, we first have to know how animals resist disease in general. Our immune system has evolved to defend against invading bacteria, viruses, and other smaller organisms, also known as pathogens. It's an extremely complex process with many moving parts that people have dedicated their lives to understanding. But from what we do know, certain parts of the immune system are common across species, including us humans. Typically, our immune system has B cells that are always on the lookout for invading pathogens. Each B cell produced in our bone marrow has a unique receptor on its surface. When a B cell is sent out into the body, it is on the lookout for pathogens that match that receptor. Once they find one, the B cell is activated and will remember that specific pathogen. The B cell then divides into thousands of B cells or plasma cells. B cells will be inactivated until they come across the same pathogen again. Activated B cells can also divide into plasma cells. Plasma cells produce lots of little proteins called antibodies. Antibodies have many uses in our immune system. They are basically used as weapons by our plasma cells. Antibodies will bind to pathogens they've been designed to recognize. This keeps pathogens from infecting us and doing harm. Binding also helps recruit other cells in our immune system that are specialized to tear down pathogens. Finally, antibodies will also cause inflammation, which stops further infection. Our bodies are always producing antibodies that will help get rid of any pathogens they find. This is known as our humoral or passive immunity. But sometimes pathogens attack our bodies in large numbers or come across new pathogens that we don't have antibodies for. In cases like these, the antibodies we already have are not enough, and the plasma cells have to produce more to be able to defend against these pathogens. When our body has to produce more antibodies to fight off pathogens, it is called active immunity. As you can see, antibodies are very important in our body's immune system. Without them, we wouldn't be able to defend against harmful pathogens that could cause disease or infection. In fact, most if not all vertebrates have antibodies, including sharks. Of course, antibodies between species have to be different since the same pathogens that can infect a shark might not be a threat to us. But in general, most antibodies look like this. This is the human antibody. There are many different kinds of antibodies. Some can be longer, or some can look like many attached together. This antibody is made of 4 parts, but since it's symmetrical, there are only 2 distinct pieces we need to worry about. The first is the heavy chain, named such because it's twice as heavy as the light chain. The heavy chain is also called the constant domain because the amino acids or building molecules that make it up are pretty consistent between individual antibodies. However, the length can change depending on what kind of antibody it is. The heavy chains will cooperate with the light chains to help the antibody bind to a pathogen. Light chains are typically smaller than the heavy chains, but they also have a different function. The light chain is also called the variable domain. This is because it will be different between antibodies based on the plasma cell that makes it. These light chains are different so that they can recognize different pathogens based on the shape of certain structures on the pathogens. For example, the COVID-19 virus is well known for its spike proteins that cover its surface. Antibodies that can defend against this virus would need to have light chains that are complementary to these spike proteins and are designed to stick to them with temporary opposite charges, bonds, or even similar properties. Many viruses have protein structures on the outside of them, so our bodies need many different antibodies that can match these protein structures. The variable domains on antibodies can match these protein structures like puzzle pieces, but they can also match them based on the similar forces between them, almost like adhesives. The heavy chains and light chains are held together by disulfide bonds. In a disulfide bond, 2 sulfurs stick out of each chain and share electrons, which holds them together. There are also disulfide bonds in the hinge region, which keep the antibodies flexible. Since disulfide bonds are covalent, or share electrons, they are much stronger than other bonds that hold antibodies together. And that's the human antibody, which shares a lot of similarities with other antibodies in vertebrates. But of course, if different animals need different antibodies, their antibodies are going to look different than ours. This includes sharks, which look like this. As you can see, their heavy chains are much longer than ours. In fact, the shark antibody is only heavy chains. This is because all sharks live in salt water, and just like eating salty foods, living in salty waters puts the organisms that live in it in danger of dehydrating. Basically, water will want to leave the cells with a low amount of salt in them and go to the ocean, which has a higher amount of salt. Without water, cells wouldn't be able to function. To keep water from leaving their cells, sharks produce large amounts of the chemical compound urea. In simple terms, shark cells keep the amount of urea in them equal to the amount of salt in the ocean, which keeps the water from leaving. The production of urea keeps the sharks hydrated, but it also introduces a problem. Urea is a strong denaturing agent to many different kinds of proteins in the cells, including antibodies. Most proteins are made of organic molecules called amino acids. These amino acids bond together and form long chains. Based on the amino acids in these chains, the chain will fold into shapes like antibodies. These shapes are held together by forces and bonds similar to the forces that will bind an antibody to a pathogen. Urea will get in between these forces and will make the protein unfold. If something like an antibody were to unfold, then it wouldn't be able to bind to and stop pathogens. By only having heavy chains, shark antibodies aren't as affected by urea. In addition, they are folded together with more disulfide bonds than heavy chains in other animals, making urea even less likely to disrupt them. But if sharks don't have light chains, then how are they able to bind to pathogens and recognize them? Well, you would think that only having heavy chains on their antibodies would make them perform worse, but in actuality, no light chains means that the binding region on each antibody is smaller than the regions on our antibodies. Since this region is smaller, these antibodies are able to reach parts of pathogens that ours can't. They can actually bind better to pathogens. Remember those spike proteins on the COVID-19 virus? Well, part of the issue of COVID-19 is that these spike proteins are constantly changing with each evolution of the virus. This makes it hard to defend against since new antibodies need to be made as fast as the virus evolves. But shark antibodies are different. Researchers have found that shark immunity against COVID is just as effective as the immunity of people who have already gotten the virus and have produced the antibodies to fight against it. Since shark antibodies are so small, they don't bind to the spike proteins and instead bind to a region of the virus that isn't changing as the virus evolves. As long as these antibodies can recognize that region, then COVID isn't a problem. We may not be able to use shark antibodies for our own immune systems, but they still hold a lot of value for the medical community. Since the antibodies are so good at targeting and binding, they can be used for a variety of purposes. Researchers and companies have seen their potential in developing treatments for cancers and autoimmune diseases. Besides treatments, they can also be used as databases for molecular structures and in turn for diagnosing diseases. More mundanely, they can also be used to extract target proteins from serums. Shark antibodies' unique properties give it a lot of potential for medical technology. If one animal is capable of so much, then imagine what other animals or organisms could be hiding. By exploring and researching Earth's organisms, we can not only learn more about them, but also ourselves.